we got this one relocalized away from the nucleus. But unlike dietary restriction, we found that this was not dependent on this protein RAD53. And that was sort of interesting that we got separation of function between TOR1 and dietary restriction. And most people can't just delete TOR in their cells. It's a very essential protein. These are a little bit special. So we want to know if rapamycin had the same effect. And indeed it does. It relocalizes LOS1, just like dietary restriction. But it does it in a RAD53 independent manner. And something that was sort of parallel to RAD53 and not directly upstream of it, we would expect that if we delete TOR1 in a RAD53 cell, it should still be able to extend lifespan. And in fact, it does. So it was really cool that we were starting to build up really common dietary restriction mimetics and forming a pathway where tRNA was at the center of it, all on this side of this diagram. And while that was cool, we wanted to know was downstream of this nuclear tRNA. If tRNA wasn't just acting in translation, it was actually acting as a signal, what could that signal be? We have a little bit of background data that GCM4 may be involved. This is something that was found in the same reduction and translation paper, where if you delete this transcription factor, you no longer get a full lifespan extension by things that are thought to be in the dietary restriction pathway. But you still got a change in lifespan that was about 30% in all these cases. So it was very clear that something else was involved in dietary restriction. There was a big gap in the story for what was actually causing things to be long-lived by dietary restriction. I wanted to go after that and figure out what it was. And in fact, in LOS1, we checked that GCM4 deletion has this sort of same effect as these other DR mimetic mutants. It doesn't completely reduce lifespan, but it has this sort of partial effect. And again, Matt let me play with a microarray to sort of go after this question. And we got something which at first I was a little bit downtrodden because we only got about 1% of the genome that was upregulated in a LOS1 deletion mutant. But then in hindsight, I thought this really narrowed down what actually might be important in lifespan extension. I thought anything that was upregulated might be causal for the lifespan extension. And when we look at these 60 genes, we found that 88% of them were GCM4 targets. So that was sort of promising that was in this DR pathway, since this is the only other thing that's really been shown to be downstream of it. But it wasn't the whole story. So I want to look at other transcription factors which might be involved in lifespan extension as well. And eventually I started focusing on this protein called GLN3. And GLN3 regulates only 13 different genes that were upregulated in this LOS1 deletion mutant. And they're all involved in sort of amino acid biogenesis. I didn't really understand that. But at this point, I said, okay, I have a transcription factor to go after. Is this going to be sort of the DR transcription factor? And in fact, it was. It was a really important result. The first thing that happened with it was we overexpressed GLN3, and we found that if you get more quantity of this transcription factor in the cell, you get a huge lifespan extension. This was about 80%. And sort of to build up and make sure this was what we thought was actually happening, we could go after different genetic bases to see if we got a similar result based on what should upregulate GLN3. So URI2 is something that holds GLN3 in the cytoplasm, so it can no longer go to the nucleus and activate transcription. If you get rid of that thing that's holding it in the cytoplasm, you also get a lifespan extension. And similarly, there's a sort of a physical blocking protein that binds to genes. That's called DAL80, and this one binds to the genes that GLN3 activates. So if you get rid of that block, you should still get more expression of those genes. And in fact, we got a lifespan extension. So it's very clear that GLN3 was sufficient for lifespan extension, which is cool, but not necessarily super novel because so many genes are starting to be shown to have that effect. But what was really critical was placing it directly in this dietary restriction pathway. And probably the coolest panel in any figure of the paper was that we could delete GLN3 in this really dietary restriction sensitive background, this 3251 background. It gets a huge lifespan extension normally from 30 to 44 in this case, but without GLN3, it blocks 100% of the lifespan extension. And this was very different from what we had seen for GCN4. The only other transcription factor really shown in these to be modulating lifespan extension by dietary restriction. That one only reduced it by about 30%. This was 100%. So this was, in my mind, the transcription factor for dietary restriction. It was a really cool result. And at some point, you want to get at some sort of mechanism for why these yeasts are extending lifespan. And since we had already talked about RAD53 and DNA damage sensing, I figured that, well, certainly these proteins may be involved in that process as well. And so we took a LOS1 deletion and a TOR1 deletion and hit them with the same chemical that induces DNA damage. And this normally kills off about 50% of yeast. Only 50% of yeast can form colonies after that. But if we delete these genes, which when they're deleted cause longevity, it also causes these cells to, once again, survive despite this damage. And in these cases, if you could remove GLN3, this transcription factor that we thought was necessary for lifespan extension by dietary restriction, we no longer had that sort of suppression effect. So this is really promising to me that it could be because of reduced rates of DNA damage that these cells were able to live longer. And similarly, if we overexpress parts of the pathway over here that could extend lifespan, we got the same sort of DNA damage resistant effect. So that was really cool. So sort of the summary of this project was a lot of arrows in all kinds of different directions with lots of different proteins. But in the end, the core results of this project, which were completely novel and interesting, was that tRNA might not only act as something involved in translation. It might act as something that actually signals to the rest of the cell for stress resistance. And that was a really key finding. And beyond that, we had been searching for a very long time what was sort of downstream of dietary restriction. And we finally got it down to one transcription factor, and that was GLN3. So those two results are something that I hope you'll take home today and think that's really interesting and worth studying. So in the end, just to summarize my PhD, yeast lifespans are really cool. And that's why I keep nerdily talking about them, despite whether or not you guys care about it. It allows for a sort of genome-wide understanding of longevity pathways, and that's why we went over that evolutionary question. That in fact, yeast seem to follow this reasoning that if you reduce fecundity or increase fecundity early in life, and that is something that yeast are very selected for, it might make it so that they're not able to live as long. And that seemed widely true. And then we went after sort of molecular pathways for why yeast are long-lived. We spent a lot of time just showing that sirtuins are bunk. They don't really do anything that's very interesting in yeast, maybe SIRT6 in mice. But more importantly, I came up with this really cool project that nuclear tRNA could regulate far more than we ever imagined. And I thought that was just a really cool find. And so with that, I must have been talking really fast because I finished earlier than I expected. I want to thank everybody pretty much everybody in this room has helped me out at one time or another. You guys are great. I love this picture because you guys are happy. You guys are the reason I come into lab every day, really early. And, there's a ton of dissectors who have helped us get these lifespan data, and they've been completely integral to this process. And Daniel Carr has been really helpful in making sure they come into work on time and know what they're doing. Thank you very much, Daniel. Chris Murakami, that's a really interesting guy here. He helped me get into the lab. He was really the lab manager for many years, and he was one of the initial people that made me think that okay, yeast research is really interesting. I should go after this. Brady Elson, just for the quote that science is cool. Brady's done a ton of work to make us all more efficient. He's created our databases and helped us with lifespan analysis. It's been really great working with him. George Sutphin, he helped me out a lot in that SIRT2 project. Thank you very much. And sort of my core team here of the undergrads who work with me, Amanda Almond and Sylvia Sim, who regretfully couldn't be here today because they're often doing better things. And Annie Zoe, who's, as you saw, part of lab drama, because she's my girlfriend and has helped me out very much. Hopefully I've helped her out a little bit too. And of course, I couldn't have done any of this without Matt. And just I want to thank him for making this such a rewarding experience and the cave behind that. So with that, I'll take any questions.
Questions?
If I get it. Yeah.
Is this the last one? Is that specific for yeast? The exporter or worms have to? And if you tried siRNA,
almost every single part of this pathway is conserved, with a single exception of GLN3. So Los one, there is a homolog in worms, and we have actually tried to see if it's long-lived when we reduce its function, and unfortunately we didn't get the cool result. But there is an export in worms as well, and people have shown in mammalian cells that this is the protein that actually binds directly to tRNA and can shuttle it between the nucleus and the cytoplasm. So most of the experiments have actually been done in mammalian cells.
Is that the only one in mammalian cells, or there are redundancies such that
Right.
the worms you might not have expected in your siRNA if there was a second?
Right. I'm not really sure how many different homologs there are in worms. Worms is when I blasted most parts of this pathway against worms, mice, and humans, worms actually had the least conservation in most of these different pathways, which is a little refreshing because that's a lot of what the lab worked on. We could have done a lot more interesting research on. But it really was hard to look at what to go after. But there's certainly redundancy. So if you get rid of this exporter, they're still alive. In fact, they grow completely normally. So it's interesting to figure out what these different things are. In the Hopper Lab's been trying it for 30 years. Just can't figure it out. Yeah, Alan.
Do you think that the effects might be mediated by just one or two tRNAs, or is it?
That's a really interesting question. So there's only sort of two tRNAs that were generally looked at between this dietary restriction process. So they always did fish hybridization assays to see if it was going to the nucleus. And they tried one tRNA that had an intron in it, and I forget which one that was. It was a his one. And they tried one that didn't have an intron in it, and they showed in both cases whether or not it had this intron, they were relocalizing to the nucleus under dietary restriction. So it seems to be a general phenomenon. And I think the current understanding is whether or not it's amino-acylated in the nucleus is what finally determines whether or not it gets put out to translation.
Do you ever try any binding studies where you were take protein, mix tRNA together, see if you can pull it down or something like that?
So you're thinking what else could be acting as Los one, something
Yeah.
like that. And I feel like somebody must have tried this at some point. But you would think if you take tRNA and pull down things, you're just going to get ribosomes, right? And I suspect that must be what people have done. But I haven't seen any data for it, probably because nothing ever came out of it. But I think it's an interesting question, definitely. Yeah.
If you continue with this project, what would be like your next best step?
Next best step. So Mitchell's rotating our lab. Yeah, obviously a very, very interesting question, and it's something that the reviewers brought up when we first submitted this paper. Is that it's really cool, but they said that this was just one more long-lived gene in hundreds that we know about. That was the Los one phenotype, and that you just end up with the transcription factor. And I think the transcription factor is really interesting because it's something that you can regulate and sort of target. But they thought in terms of figuring out what was causing aging itself, it wasn't very informative. So what they wanted was to look more downstream of that transcription factor. And that's actually what you and I have been doing is creating mutants for those 13 different target genes and seeing their lifespan effects. And I personally think it has to do with this. It does certainly tie into amino acid biosynthesis and eventually create some sort of signaling event. And whether or not it's nitric oxide or not, we'll find out. So nitric oxide is sort of a byproduct as well. All these enzymes are upregulated. There's some promise for why it might be involved in aging. It's involved in wound healing, and it could activate different transcription factors. But it's really not been really studied very well in the context of aging.
questions? Okay, well, we will ask everybody but the committee to leave. And there's a reception, where is that?
It's in the J K-Wing Lounge, 4th floor. Bring all the beer you want.
So, thank you, Joe.